Hello everyone, today we're going to work on photosynthesis. This is the very first topic of Unit 4 of IAL and Edexcel biology. The very first thing I will do here is to write the equation of photosynthesis. So I'll start with the raw materials or the reactants. That's when carbon dioxide with water from the soil combine with the help of chlorophyll and sunlight. So sunlight provides the energy, and chlorophyll here is the one required to capture the light energy. Together, the carbon dioxide and water will produce glucose. That's the main product. Plus, we make a byproduct of photosynthesis and the gas oxygen. We're going to have a closer look at photosynthesis. So we're going to look at the equation we've just studied. We're going to look at it in full details. First of all, water comes from the ground by osmosis. So the way water gets into the plant roots is by the process of osmosis. It travels all the way up through the special vessels. You've probably heard about xylem. So those xylem vessels is where water travels. So note here in blue, those are the xylem vessels. And here on the right is where we have the phloem. Phloem is what transports the products of photosynthesis. As the water gets to the leaf, it would combine with carbon dioxide. Carbon dioxide enters the leaf by diffusion, since it's a gas, and we get that two products of photosynthesis. The first product of photosynthesis, which really matters to us, is glucose. I'm going to write the formula of glucose, C6H12O6. So that glucose we've just made by photosynthesis, as you can see, it's made up of carbon. This carbon is from carbon dioxide. There is a hydrogen. The hydrogen comes entirely from water, and there is oxygen. Now, the oxygen, you have to realize that the oxygen in glucose comes from the carbon dioxide. So that's like an important fact here, that the oxygen that comes in the glucose formula, that oxygen comes entirely from carbon dioxide, because the oxygen from water is basically lost to the external environment, right? So again, there's carbon, there's hydrogen. The carbon comes from carbon dioxide, the hydrogen comes entirely from water, but the oxygen here, the source of oxygen, is going to be carbon dioxide. What about the oxygen? Where does the oxygen come from? The oxygen, as the product of photosynthesis, this oxygen is going to be produced by the water. When you split the water during the very first stage of photosynthesis, that's where we make this waste product oxygen. We could actually observe oxygen produced by photosynthesis. If we take any water plant, we call this the pondweed, and we collect the oxygen as we expose the plant to the light. And as you can see, there are bubbles of oxygen, and these bubbles are the ones made by the splitting of water. We call this action of splitting of water, we call it photolysis. What's going to happen to glucose, the second product of photosynthesis? So there are three possible destinations. Glucose could possibly be used straight away as a source of energy for respiration. It could be transported around the plant in the form of sucrose. So the sugar transported in phloem is known as sucrose. And the last possibility is to store that sugar in the form of starch. So plants have storage organs such as those found in the roots, and there we could keep that glucose for winter in the form of starch. In case you're using my textbook, The Complete Notes for A2 Biology, we're covering the very first topic, which is photosynthesis. That's the first topic of Unit 4. We're not going to cover the entire topic today because it's quite long. So we're going to cover the first part of photosynthesis. So as you can see here in the notes, you have all the little details about photosynthesis. It's written in a way that makes it very easy for you to remember the key points, the key words. Those are the ones that really matter when you're writing long answers in the mark scheme. You also have the important practical. So every time you have this sign, this means there is a practical, and today's practical is going to be the separation of the pigment of photosynthesis. Now that we've learned about the basics of photosynthesis, the syllabus wanted us to learn about ATP. What is ATP? ATP is like the energy currency of the cell. It can exist in two forms. ADP, with D for diphosphate, that's the form where it's uncharged, but it also exists in the charged form. The charged form is the actual ATP. Now, it's important here to learn the chemistry of ATP to understand how we could possibly charge or uncharge the ATP. First component of the ATP is the sugar. So we have the sugar known as the pentose sugar. That's the same sugar you have in DNA and RNA. To the first carbon of the sugar here, we have the adenine group attached. So that's A for adenine, and that's the sugar. We also have another component, actually 3 groups of phosphate are attached here. So we have 1, 2, 3 phosphate groups. All of these are attached to the 5th carbon of the sugar. And what really matters here is this last bond with the last sugar, with the last phosphate, I should say, here on the left. This last bond could possibly be broken down. When I break down this bond, this means I'm releasing energy. But when I make a new bond between the last 2 phosphate groups, that's when I'm storing energy. So once again, ATP is the energy currency of the cell. It can exist in 2 forms. A form that is fully charged, that's the actual ATP, and the form that is uncharged, which is known as the ATP. So when I am going from ADP to ATP, that's where I'm storing energy. That's when I'm making a bond. So that's a condensation reaction, combining this phosphate here and making the ATP. But I could also hydrolyze this last phosphate group, release energy, and that energy can be used for plant growth. It's important to understand here that photosynthesis indirectly provides ATP for the plant. Now, how does this happen? Think about it that way. The light energy first is captured by the chlorophyll. Photosynthesis produces chemical energy in the form of glucose, and the glucose can be used for so many different uses in plants, including the storing of starch, the making of cellulose in the cell walls. Glucose also is important for the making of amino acids. That's when we combine glucose with nitrates from the soil. We could also make lipids. So glucose can also be stored in the form of lipids. But what really matters for us now is that the same glucose can be used for respiration. When the plant respires the glucose in the mitochondria, it will produce ATP. And from here, that ATP we've made can be used for the different functions in the plant, including growth. In order to understand photosynthesis, you really need to have basic knowledge of chemistry. The important part of chemistry you need to learn here about is what we call redox. Redox is 2 processes, reduction and oxidation. Now, redox or oxidation reduction must always occur at the same time. In biology, we only focus on the hydrogen part of redox. So when one chemical loses hydrogen, this means that this chemical is basically getting oxidized. But when another chemical gains that hydrogen that was originally lost, it's going to get reduced. Now, the hydrogen atom is not going to fly like this in the cell because we have specific carriers for hydrogen in the cell. These carriers, we usually call them coenzymes. A coenzyme is simply a chemical that helps enzyme reactions. We have an important coenzyme for photosynthesis. We call it NADP. You don't need to know the individual meaning of each one of those letters. You can write NADP in the exam. So that NADP basically functions as a carrier where it takes this hydrogen from one chemical and dumps it on the other chemical. So the NADP exists in 2 forms. One form is the reduced form. The reduced form is the one that is attached to a hydrogen. So that's why we call it NADPH. So that hydrogen can be donated to another chemical, so then it becomes NADP, and that's what we call the oxidized form of the chemical. So again, that NADP is responsible for reducing one component of the cell and oxidizing the other. In our case, the chemical that gets reduced in this case is going to be carbon dioxide. So when carbon dioxide takes hydrogen, it turns into glucose. And the other molecule that loses hydrogen is water. So basically, these hydrogens you have here in water, they are taken by the NADP and they're dumped on carbon dioxide in order to form glucose. So that's the redox part of photosynthesis. We're back to the syllabus. They want us to learn about the structure of the leaf and the chloroplast. So there are 4 main adaptations of the leaf, including the fact that leaves are thin, that allows short diffusion distance. Leaves are very broad compared to the thickness that allows absorption of more sunlight energy for photosynthesis. Leaves contain veins which are rich with xylem, and that allows both the transport of water and provides support to keep the leaf exposed to the light. Leaves have tiny pores, we call them stomata, and stomata is where gas exchange takes place. In order to understand the process of photosynthesis, you need to have good knowledge of the leaf structure. Now, you've probably done this in your IGCSE, but I'd like to remind you about the structure of the leaf. We have 2 layers here. We call them the upper and the lower epidermis. Now, both of these cells are transparent. They allow light to get into the leaf. In between those upper and lower epidermis, we have the palisade cells. Those are very rich with chloroplasts, and that's where most photosynthesis happens. The spongy mesophyll are less rich with chloroplasts, but that's where gas exchange mainly happens. Right on top of the upper epidermis, we have the waxy layer or the waxy cuticle. This layer is to retain water inside the leaf. We do also have stomata. Those are found mainly the lower side of the leaf, and that's where gas exchange takes place. During the times of light exposure, you'll find that carbon dioxide enters the leaf because it's a raw material of photosynthesis. Oxygen is produced by photosynthesis, so the gas diffuses out. Also, during photosynthesis, the plants lose water. That's known as transpiration. Now, the loss of water by the plant is a consequence of photosynthesis. The plant can't help not to lose water, although it's an unfavorable process. Always important to practice exam questions while you're learning, after you're learning, before the exam. Practicing past exam questions is vital to score a good grade on the exam. Now, I'm taking this question from my past paper book. So we have the classified questions. I've divided this book into several chapters, and each chapter has questions only related to the lesson that you're learning. You do have a book for Unit 4, you have another one for Unit 5 and Unit 6. So each one of those books contains the questions, but you do also have the answers, which is really helpful. I've written all of the answers you need in a way that makes it very easy for you to understand, because if you just look at the mark scheme, you may not understand all the points. So I've just written all of the answers in a good way. I've highlighted the key points that you need to learn and write in your exam questions. Let's take a look at this question. It says, explain why leaves got a thin lamina. They've provided us with the diagram or the picture of the leaf here, showing us the different parts of the leaf. So the lamina is basically the leaf blade. And they also ask us here, why do the leaves contain a midrib or veins? So let's start with the lamina. What's the point of having a thin lamina of the leaves? The leaf blade or the lamina is thin because this allows short diffusion distance of gases. Remember, the shorter the distance, the faster the diffusion of gases. Now you need to tell here why carbon dioxide is needed for photosynthesis. Carbon dioxide is needed for what is known as the light-independent reactions of photosynthesis. I'm going to tell you more about it in just a moment. The lamina also allows the light to penetrate. So basically the blade or the leaf blade is needed for both the diffusion of gases being thin and being wide or broad that allows more light energy to be trapped. What about the midrib or the veins of the leaf? Those, as I told you, those are rich with xylem, but they also contain phloem. Now, xylem is to transport water. Water is a raw material for photosynthesis, but they also contain phloem. Now, phloem is important to carry the products of photosynthesis. Remember, the product of photosynthesis is glucose, but what gets transported in phloem is actually sucrose. We're going to look at the structure of the chloroplast. That's where photosynthesis happens. Now, we could divide the chloroplast into two main components. One is going to be the thylakoid and the second is going to be the stroma. Now, let's take a look at the structure in full detail. So what I have here is the stroma first. So that's the liquid or the fluid part of the chloroplast. I do have those stacks of membranes. So those are called the thylakoid. Each is called the thylakoid. And connecting those thylakoids together, we have these filaments, or we call them the lamella. And those lamellae are the ones that link up the thylakoids and put them in the right orientation. It's like the perfect orientation to receive as much light as possible. In addition to that, we do have the outer membrane and the inner membrane. So again, the chloroplast looks something like this. I always think of it as if it's like a stack of membranes. Each one of those membranes is known as the thylakoid. The entire stack is what we call a granum. The fluid part, which is like quite similar to the cytoplasm of the cell, because the chloroplast used to be a prokaryotic organism, like a bacterium that is photosynthetic. And we have those membranes, or the connections of membranes, we call them the lamellae. Those are the ones that connect the thylakoids together. So chloroplast is so much adapted for photosynthesis. You can see here we have all of the thylakoids shown as stacks of membranes. We have small ribosomes within the chloroplast, and we have here shown in white, we have the lamellae. Where is the evidence of the prokaryotic origin of the chloroplast? So what I have here is like a primitive cell, the original eukaryotic cell. This cell has engulfed a bacterium or a prokaryotic organism. Now, this prokaryotic organism turned out to be photosynthetic, meaning it contains chlorophyll. Now, as you can see, the endocytosis of this bacterium ended up forming two membranes, because the first membrane is the one that belongs to the vesicle of the cell, and the second membrane is the actual membrane of the bacterium. Now, together, those two cells, a prokaryotic, smaller one, and an eukaryotic, both cells are benefiting because the prokaryotic cell, which is turning into a chloroplast, is providing glucose, and the eukaryotic cell is providing the raw materials for photosynthesis. This became later on the current photosynthetic plant cells with the chloroplast inside of it. Now again, if you've been asked about the evidence of the prokaryotic origin of the chloroplast, you can mention all of these things. So first thing is going to be small ribosomes. So the ribosomes are 70S ribosomes, or you can call them small ribosomes. We do have the circular DNA. So the chloroplast comes with its own DNA that codes for its own enzymes, plus this second membrane, because the fact that the chloroplast has two membranes, inner membrane and outer membrane, this all tells you that this used to be a prokaryotic cell. Here is a chloroplast. So what we're going to try to do here, we're going to try to match each one of those functions at the bottom to the structure of the chloroplast. So maybe you can pause the screen and try to do this yourselves. Now, first thing, synthesis of enzyme. This means they're talking here about protein synthesis. So the chloroplast makes its own proteins with its own 70S or small ribosomes. It says here partially permeable. This means we're talking about membrane. So we do have the outer or inner membrane. Both are partially permeable. These are the ones that would allow the raw materials of photosynthesis to enter and products of photosynthesis to exit. Now it says light-dependent reaction. This means it's the part of photosynthesis that relies on light. I'm going to tell you more about it in just a moment. So that means we're going to point at the thylakoid because that's where chlorophyll is found. And we can also point at D. So it could be either D, a single thylakoid, or a stack of thylakoid. That's what we call a granum. Both would be a correct answer here. It says stores genetic information. This means it's DNA. The chloroplast does contain circular DNA. That's the one that stores genetic information for the chloroplast itself. It says carbon fixation. This means we're talking about the light-independent reaction. And the light-independent reaction of photosynthesis means the reactions of photosynthesis that don't rely on light. Those take place in the stroma. So you can see there's a small arrow F pointing here at the stroma. Finally, it says align the thylakoid to absorb as much sunlight as possible. Here we're talking about the lamellae. Those are the membranes that connect the grana or the thylakoid together, and you can see it here shown as part or label. Now, we need to learn about the different pigments that plants have. Now, chlorophyll is not like a single pigment. Chlorophyll is actually a mixture of different pigments. Each one of those pigments absorb different wavelengths of light. In the syllabus, they want you to understand two terms. One is called the absorption spectrum, and one is called the action spectrum. In addition, they want you to learn how to separate the different pigments of photosynthesis. The information about the action spectrum is found in your textbook, the complete notes. It's found on page 3, and the practical related to this part of the syllabus is found also on the same page. To understand what is meant by an action spectrum, it's good to try an experiment like the one I have. So I have a light source, I have filter, and I have a plant here, water plant, so I can measure the volume of oxygen produced over units of time. This will help me to find the rate of photosynthesis. So let's start by exposing the plant to the light. As you expose the plant to the light, you notice there are bubbles of oxygen, and I'm measuring the volume using a gas syringe, and I'm measuring the time it takes for the volume to be produced. Now, as you can see, different colors of light allow the plant to do different rates of photosynthesis. So you can see that purple blue do actually allow the plant to do a lot of photosynthesis. But when the light turns green, the rate of photosynthesis gets slower, then it gets better with yellow, and it gets better and better with orange. As you can see, different wavelengths do allow the plant to do photosynthesis at different rates. So that's what's basically meant by an action spectrum. So this is a typical action spectrum graph, and as you can see here, it tells you where exactly the plant does a lot of photosynthesis. But why is it that plants do prefer different wavelengths of light? This has to do with the pigments that plants have. Now, as I told you, there are three main pigments that plants have. There is two forms of chlorophyll, each absorbs different wavelengths of light. In addition, we have accessory pigments known as the carotenoids. Those carotenoids, as the name suggests, has orange color, and we do actually see those pigments quite well during autumn because chlorophyll is broken down during that season as the time of the day or the exposure of the light decreases, and you start to see those orange pigments known as the carotenoids. So those carotenoids basically allow the plant to absorb different wavelengths of light because they have different colors. You can see here in the absorption spectrum, we can see that there are three different pigments, the one I've just told you about. You can see that the chlorophyll, although they're both called chlorophyll, but each version of chlorophyll, A or B, absorbs different wavelengths of light, and the carotenoids do absorb different wavelengths of light. Again, you have to keep something in mind here, that the color of the pigment suggests that's the color that is reflected by the pigment. So again, if the pigment's color is orange like those carotenoids, this means they do not absorb that orange color. And if chlorophyll color is green, this means it doesn't absorb green. One practical we do in this lesson is to separate those different pigments that plants have to absorb light. So the method is quite simple. First of all, we're going to collect leaves, we're going to crush them, dissolve them, and then we separate them by filtration. So again, the very first thing to do here is to crush leaves that are rich with pigments such as spinach. And then as we crush them, we need to add some sand to provide some rough surface. We dissolve the pigment in a suitable solvent like acetone or ethanol, and then we separate those pigments by means of filtration. So we have now like a paste of all of those pigments. We can separate them, and then we apply those pigments to the chromatography paper. It's important here to draw a line on the chromatogram or the chromatography paper. We draw this line using a pencil because the graphite or the pencil doesn't dissolve in any pigment. So now that we drew that line, we call that the baseline, we take those pigments and we put a drop of those pigments on that baseline. Right after this, we take that chromatography paper and we put it into the solvent. You have to be careful. You shouldn't let that pigment dissolve in the solvent. So you just have to keep that line above the level of the solvent. The end product is going to look something like this. So you have now the pigments been separated, and in order to identify each and every one of those pigments, we measure what we call the RF values. What's an RF value? RF value is simply the distance traveled by the solvent, which is always going to be greater than the distance traveled by the pigment. So we just divide that distance here, like let's say the distance traveled by the pigment is like almost here 3.8, and the distance traveled by the solvent is nearly 4.9. That would be the RF value of this particular pigment, and if we could just look up this RF value in data books, we could identify this pigment. We're going to look at a new term. This is known as compartmentalization. Compartmentalization is the fact that the chloroplast is divided into two main parts. You have the thylakoids. That's where the light-dependent reactions take place. Those are the reactions that directly require light to take place. And you have the light-independent reactions that takes place in the stroma. So those light-independent reactions do not directly need light, so they can still occur day or night. Let's differentiate between the light-dependent reactions and the light-independent reactions. During the light-dependent reactions, we're going to use light energy to break down the water molecule. This reaction is known as photolysis. Photolysis stands for the fact that light is going to be used to break down water into three main components. You have protons electrons. Those are the parts of the hydrogen atom, in addition to oxygen. Oxygen is a waste product that will soon diffuse out of the chloroplast and out of the plant. But what are we going to do with the hydrogen atoms? We're going to carry those hydrogen atoms with the help of the NADP. So the NADP will get reduced, carrying those hydrogen atoms from the thylakoid all the way to the stroma. In addition, we're going to use those hydrogen ions, we're going to use them to generate ATP. So we're also going to make ATP with those light-dependent reactions. In the stroma where the light-independent reactions take place, we're going to take the products of the light reactions, which are the ATP and the reduced NADP. We're going to use them to fix carbon dioxide. So carbon dioxide from the atmosphere can combine with hydrogen coming from the water originally. We're going to combine them to make glucose. This reaction requires ATP. So energy from ATP is used here for the carbon fixation. Right after this, that ATP turns into ADP, and the carbon dioxide is no longer there because it's entirely turned into glucose. So the ADP that formed with the light independent reactions will return back to the thylakoid, where it will be recycled into ATP. Let's summarize the reactions of photosynthesis. We're going to start with the light-dependent reactions. Those are the ones that take place on the thylakoids. So first thing, water comes into the plant. The water is going to be split by light. It's known as photolysis. As a result, the water will be broken down into oxygen, which is a waste product, but we're also going to make two useful things. One of which is going to be ATP, and the second is going to be reduced and ADP. Together, those, the ATP and the reducing power from the hydrogen, going to be used to fix carbon dioxide. So carbon dioxide from the atmosphere is going to be fixed with the addition of hydrogen to form glucose, and the products now of the light independent reactions going to return back to the light reaction. So basically we're going to recycle the ATP and we're going to recycle the NADPH. So ADP and NADP will return back to the light reactions. So this is why those two reactions of photosynthesis, the light dependent and the light independent, are so much interconnected. One of those reactions cannot occur without the other. We're going to differentiate between the light reactions and the light independent reactions using this diagram. So pause the screen and try to find which one of those properties belong to the light reactions or the light independent reaction. So let's start with the photolysis, or the splitting of water. This takes place in the light reactions of photosynthesis. Also, the ATP is made in the light reaction. However, the hydrolysis, or the breaking down of ATP to ADP, is made in the light independent reaction. The light independent reactions take place in the stroma, and that's where carbon fixation takes place, while the light dependent reactions occur on the thylakoids. The making of glucose is part of the light independent reactions, and the reduction of NADP takes place in the light dependent reactions, but the oxidation is occurring in the light independent reaction. Here is a sample exam questions. They've given us the envelope of the chloroplast, which is the double membrane of this organelle, and they wanted us to draw the parts of the light dependent reaction. So we're going to have to draw every structure related to this reaction. So basically all we're going to have to do here is to draw thylakoids, or like a stack of thylakoids, like what I'm drawing here. And you just have to label those. So we're going to score 2 marks if we just label the thylakoid, and I'm also going to label the stack. So the entire stack of thylakoid is what we call a granum. We're halfway through the topic of photosynthesis, so we still have to study the reactions of photosynthesis in full details. Now, if you still need the remaining part, in addition to the other parts of the Edexcel syllabus, you can go to my website. I'm leaving a link in the description. In my website, you can find the notes, like the ones I was showing you at the beginning of the class. You can find solved questions, in addition to videos, just like this one, for every single topic of Edexcel AL biology.